चलिए हम स्टार्ट करते हैं. Can you see the screen, guys? Kina and Naila दोनों.
जी सर.
Yes, sir.
ठीक. So structure of amino acids. Amino acids are the building blocks of proteins. Okay. The basic structure is as follows. This is carbon atom. Central carbon atom होता है. And a carbon atom can have a maximum of 4 bonds. Okay. Due to its valency. One bond is bonded to the carboxylic acid group COOH. Other one is bonded to the amino group NH2. Other one is bonded to a hydrogen. Last but not the least, you have an R group. Okay? This carbon atom in the center is called the alpha carbon. Okay? ये acidic group होता है, carboxylic acid का group होता है. This is carboxylic acid. This is the amino part. Hydrogen remains here, fixed hydrogen atom. And then R group or the variable group. R group is variable, meaning R group can be of multiple types. You can have acidic amino acids based on acidic R groups. You can have basic amino acids based on basic R groups. You can have polar and non-polar amino acids. Okay, so R group can be considered as the variable group, and R group provides the identity to the type of amino acid. It provides the type of amino acid. Okay, so whatever the R group is, that amino acid is going to be governed, the identity is governed by the R group. Now the smallest amino acid, now obviously you cannot learn all the amino acid names, but for your MDCAT, I would suggest you learn at least as many as possible, because they might ask you the names of amino acids on your exam. However, over here in AKU, there is a less likely chance they might ask you the names. Now this is the smallest amino acid that is out there. The R group is just a hydrogen atom. Does anyone know the name of this amino acid? It's called glycine.
Glycine.
Okay? Yep, glycine. It's a non-polar amino acid, okay? This is just one example. Similarly, you can have acidic, basic, carbo, and polar, non-polar amino acids. Now, what happens to the amino acid as it goes into the body solution? Amino acid does not remain in this particular form in the body solution, okay? When it goes into the body solution, it slightly changes. And this is going to be the new configuration. This is the amino acid that we have. It will undergo some changes in the body solution. The acidic end, because it's an acid and acids can donate protons, is going to lose a proton. Okay, it's going to lose an H positive ion. Then write the name of the title. Structure of amino acid. Okay, or just think of the amino acid to define. Oopsie. Okay. Now, this part is going to lose an H positive ion because it's the acid part, and acids donate protons. So hydrogen will be lost from here and it will be removed out in the form of H positive ion. The same H positive ion will be accepted by the basic group. This is the amino group, right? Amino group is a basic group, okay? And because amino acids contain both acidic as well as basic groups in it, we call them a special, we give them a special term. We refer to the amino acids as being amphipathic. Does anyone know what that means? Amphipathic. Amphipathic means, amphi means containing dual nature. So basically a compound that contains acidic as well as basic characteristics. Okay, amphipathic. Now the carboxylic acid part is going to donate the proton, whereas the amino part is going to accept a proton. Okay, it's going to give out its lone pair of electrons and accept a proton, because bases have the property of accepting protons, right? As a result of this proton exchange, right? It's an exchange of proton from one end to the other. You get a new species that is called, that looks like this actually. This is the carbon atom with all four bonds attached to it, but the two bonds that are on the corners, they are now in a different configuration. The carboxylic part has lost the proton, so it's gonna be, it's gonna look, it's gonna be COO negative. And the NH2 part is gonna gain a proton, so hence it's gonna be NH3 positive. Okay? But because the same compound contains positive and negative in equal quantity, so the overall charge is gonna remain 0. The rest of it stays the same. This species that has been created is called Zwitter ion. Okay, not Twitter, it's called Zwitter. Twitter se yaad kar sakte hain. Zwitter ion. And this is an isoelectric species. Okay, what exactly does isoelectric mean? Isoelectric mean having equal quantities of, iso means the same charges on both the sides. Isoelectric species has positive, equal number of positive and negative charges. Therefore, the overall charge of the Zwitter ion is still neutral. Okay, so overall neutral. Right. This was the basic structure of amino acid. And this section, the section that you guys can see on the left side, this is important for your exam. And actually quite frequently tested. Frankly, it's very, very frequently tested. Okay. Now, once you can see the amino acids over here, amino acids act as the building blocks of bigger chains called polypeptides. And those polypeptides can further give rise to proteins. So these are amino acids. Amino acids are the monomers of larger polymer chains called proteins. So amino acids, when multiple amino acids link together in a specific sequence and interact with one another, they form a large polypeptide chain. Okay. So they are the building blocks of proteins, amino acids. Now, how exactly is a peptide bond made? If this is your amino acid right over here, which there is a COOH part, the right side, and NH2 part on the left hand side, a hydrogen and an R group right over here. Let's assume there's another amino acid on the right hand side. It says carbon atom, and then it has the 4 bonds as well. But obviously the NH2 is always made on the left hand side. Okay, I'm going to draw it like this: NH2, 2 hydrogens, and then the rest of it will copy. What happens here when they two combine together? The condensation polymerization occurs. meaning the carboxyl part loses the OH and the NH2 part loses a hydrogen, forming a water molecule, and that water molecule is actually lost from here. The carbon atom on the left-hand side joins with the nitrogen atom on the right-hand side, forming a new interaction that looks like this. Carbon double bonded oxygen is bonded to the nitrogen, which has a hydrogen next to it. This is your peptide bond or amide linkage. Okay, let's write it down. Now, this bond that you guys can see over here in the center, the carbon-nitrogen bond, that's your peptide bond, okay? The entire linkage is called amide linkage. This compound that you guys can see, it has been created. It contains 2 amino acids only, has a special name. Does anyone know what I'll call this? Dipeptide. Okay. The reason why we call it dipeptide is because it's made up of 2 amino acids. Okay. Not because it has two peptide bonds. It does not have 2 peptide bonds. It only has a single peptide bond. Okay. Similarly, if you can have multiple repetition of the same procedure with hundreds of thousands of amino acids, you'll get a gigantic molecule, and that molecule would be your protein molecule. Okay, it's gonna be protein. This is how it works. Sakina, are you in class? Right? You're in class.
Yes, sir.
Take. This process that I just showed you is called condensation polymerization. Okay? Right. Now we have certain amino acids in our body. Okay? We've got 20 amino acids in our body which are being used to form proteins. In nature, you have more than 20, but the ones that we have in our body are of 20 types only. 20 different types of amino acids. Okay, these are the types, the basic types. These types, these 20 types of amino acids are subdivided into 2 categories. Does anyone know what those 2 categories are? Essential and
Essential and non-essential.
non-essential. Good. So what's the difference between essential and non-essential amino acids?
सर essential हमारी बॉडी खुद बनाती है and non-essential नहीं
उल्टा
बनाती
बोल रहे
है.
हैं. उल्टा उल्टा है उल्टा.
अच्छा.
Switch it, flip it around. So essential is not made by the body and non-essential is made by the body. Okay? Essential, therefore it's essential. So hence it has to be taken by diet. Cannot be processed by the body. Whereas non-essential, it is processed by the body. It's processed and manufactured by the body itself. So hence, no need to take it in diet. So not required in diet and essential ones are required in diet. So approximately 9 amino acids that we have are essential amino acids. But इस बुक में जो नंबर लिखा है आपने उस नंबर के साथ जाना है. इस बुक में शायद 8 दिया हुआ है. नहीं, इस बुक में शायद कोई नाम नंबर नहीं दिया हुआ. ओके, दिया हुआ है. Out of 20, the human body can synthesize only 10 amino acids का नाम दिया हुआ है यहाँ पर. 10 essential और 10 non-essential. Okay, so 10 essential amino acids are there and 10 non-essential amino acids are there. Checking guys, I don't know what's wrong here. Just keep going back. This is 10. This is also 10, okay? Right. Sakina?
Yes, sir.
Oh, acha, theek hai, theek hai, theek hai. That's it, never mind. Okay, so at least ये 20 amino acids जो है ना इनके नाम आप याद कर लीजिएगा, ठीक है? और कुछ नहीं किया जा सकता इनका, मतलब सबके नहीं तो essential के कम से कम नाम याद कर लीजिएगा, क्योंकि important है. हाँ
सर ये हमारे बुक में गिवन है.
जी जी बिल्कुल. ये सारे के सारे नहीं गिवन बट 8 amino acids गिवन है. जो बुक में गिवन है वो आपने बस याद कर लेने, ठीक है?
ओके.
Yes. Okay. Now after this we've got some reactions of amino acids and some tests for amino acids, some acidic and basic characteristics of amino acids, how they are synthesized. ये सारी चीजें आप अपने हिसाब से आप पढ़ लीजिएगा, कोई इतना इम्पोर्टेंट या अलग से कुछ इतना खास नहीं है. जैसे फॉर एग्जांपल एस्टरिफिकेशन, if we've done the previous reactions, esterification भी हमने कर लिया होगा लाजमी. Esterification, how is an ester made? When an alcohol combines with carboxylic acid, right? So similarly, when amino acids combine with an alcohol, their acidic component reacts only. Okay. Then test for amino acids. Does anyone know how do we test for amino acids? Most of the times, amino acids are colorless, especially when the sportsmen, when they are taking part in sports, and for example, if they have taken, if they have injected some illegal drugs or illegal proteins for increasing their activity during the sports, how will you detect the quantity of that illegal drug in their blood or urine? So it's by taking the urine samples and then spraying a special spray. The spray is called ninhydrin spray. Okay, ninhydrin is a special locating agent. We call it a locating agent that is used to locate the illegal proteins or amino acids. Okay, so it's a locating agent. So ninhydrin, what it does is, because amino acids are colorless, ninhydrin reacts with them and produces colored spots. All right. इसके अलावा condensation, polymerization वगैरह, polyimides are made up of amino acids, but this is all biochemistry. So you know what amides are, right? We just did amide linkage. So if you know what amide linkage is, you should know what polyamide is. Polyamide is when a compound has many amide bonds. Okay. So with this chapter is closed. Macromolecules is the next chapter, and it's not that important for me to teach you because it's all reading. Okay. And plus it will also be covered in biomolecules chapter. So we'll be doing that anyways in biology. Okay, up what I'm going to do is I'm going to start off from the beginning because we have only done organic and we have not done inorganic. Now I'm going to shift to inorganic chemistry, okay, of second year. Let me send you this PDF. नहीं गर्म है, ठंडी हो जाएगी. I've sent these notes on the group. I hope you guys received it. Okay. Up next, we're gonna start off with the new chapter. Chapter 1, grade 12 chemistry. I think periodicity है chapter. Periodic classification of elements and periodicity. Okay, let's get started.
Excuse me, sir.
जी
सर, जो रिएक्शंस हैं इनको याद करने का कोई आसान तरीका नहीं है?
देखें, इनके अपने सिर्फ रिएक्टेंट्स, प्रोडक्ट्स और कंडीशंस याद करनी है, मैकेनिज्म नहीं याद करने. बस ये एक आसानी हो सकती है. बाकी इसके अलावा याद तो करने हैं ना, रट्टा ही मानना है इसको, और तरीका नहीं हो सकता. केमिस्ट्री का ये
जी
वाला
सर.
पार्ट जो है ना, ये रट्टे वाला है. मैकेनिज्म फिर भी अगर होते तो समझने वाले होते. अब इक्वेशन को मैं क्या समझाऊं आपको, एक जनरल ट्रेंड बना बना के समझा सकता हूँ, बस मैक्स वो कर सकते हैं, इससे ज्यादा नहीं कर सकता. ठीक है.
जी सर.
तो चलिए फिर पीरियोडिक क्लासिफिकेशन पढ़ लेते हैं. हिस्टोरिक बैकग्राउंड गिवन है, सारा पढ़ना है आपने. सही है, क्योंकि इनका कोई भरोसा नहीं है, कभी कभार दे डू गेट सम सिली क्वेश्चंस related to historic background. Take a second now. Forgot to do something.